Hi guys. So we are going to do a oops. We are going to talk about the beginning of DNA, which is the genetic carrier of information. So why is it important that we talk about DNA? Well, in 2009, there was a cluster of unusual flu cases that were reported around Mexico City. And then later in that year, in June 2009, the World Health Organization, known as WHO, declared that H1N1 was essentially the first influenza pandemic, global epidemic, and it unveiled a mass effort to try to contain it. What they soon found out was that H1N1 was a hybrid flu strain. Created from a previous known flu strain, which itself originated through a combination of viruses from blood and pigs and people, mixed with an Asian swine flu virus. So essentially you had different viruses coming together. What made this combination so novel or new for the genes that were produced was that the fact that this H1N1 strain is different than a lot of flus that you knew, that it infected young, healthy people, which we're not used to seeing that. The flu usually affects the elderly or people who have already sick or compromised immune systems. So many of the countries participating in the World Health Organization coordinated a response, including widespread distribution of a new vaccine. In a typical year in the United States, more than 20,000 people died from influenza infection. And essentially, there was an H1N1 that this particular outbreak that we had is pale in comparison to the deadliest outbreak pandemic that happened in 1918 through 1919, the Spanish flu, which killed more than it took 18 months to kill about 40 million people worldwide. More people have died of AIDS since it was discovered more than 30 years, but this is more people died in just one single exposure a year, which is the reason why it was so scary. Literally, someone would be sick in your town, and within, a matter of weeks, a large portion of your town could be dead. And so it's very, very scary. The flu virus, like all viruses, consists of relatively simple structure, and we're going to talk more about viruses towards the end of the semester. But most of them have a nucleic acid. In this case, this one was an RNA and protein. Essentially, though, combating any virus requires a detailed understanding of life at its molecular level, which is one of the reasons why we're going to start talking about DNA today. Not only do we want to understand this so that we can understand how viruses work, but we want to understand DNA so that we can understand genetics and inheritance, so that we can understand cancer a little bit better when we talk about cancer coming, or what does carcinogenic mean. You might have known the case where they found a gene where, what is her name? Angelina Jolie went through a mastectomy or removed her breast because she found out that she had the gene that was essentially known to be linked to breast cancer. So being able to understand how DNA works at a molecular level is very important to be able to kind of go through life currently understanding diseases that affect our families every day. So DNA structure and replication. So we didn't know that DNA was known to be a chemical component of cells until about the late 1800s. By the late 1930s, experimental studies had convinced most biologists that one specific kind of molecule was the basis of inheritance, but we didn't know what that molecule looked like. By the 1940s, scientists knew that chromosomes consisted of basically chemicals known as DNA and protein, but we still had no idea what exactly it looked like. And so what we're going to do here at the beginning, we're going to talk about the biochemical identification of genetic material, which is AKA history. Then we're going to look at nucleic acid structure. Of course, hopefully nucleic acid structure for you is a review from Unit 3, Biomolecules. So let's talk about the history first. So genes were a mystery for a long time. Many early geneticists, which are people that study genetics or genes in DNA thought genes were proteins. Of course, we know now proteins are complex and they're very variable. Nucleic acids are much simpler molecules. So thank goodness, because it's made it easier for us to understand what's going on. Beginning in the late 1920s, evidence began to accumulate that DNA was actually the hereditary material. And through the 40s and early 50s, we were able to demonstrate what DNA actually looked like and that it was the genetic material. So let's go with the late 1920s when we talk about Frederick Griffith. He worked with Streptococcus pneumonia bacteria. And there's actually two strains of this bacteria. There's one that's called the S strain, or what looks like it's very smooth. And this one actually secretes what's known as a capsule. So essentially that helps protect this bacteria from our lymphatic system, from being able to override it and destroy it. So it's very infectious and fatal, especially in mice. The strain that doesn't have this capsule for protection is called the R strain. And this looks rough. And so it is not fatal in mice. And so essentially, just to remind you that the capsule does shield the bacteria from the immune system. So they're able to survive in the blood if you have the S strain. So that's the reason why it's essentially fatal. And so how did he figure out that? Let's just start first with the experiment that he's looking at to kind of know that something was happening, or that there were traits that could be established in a cell. So we start with the experiment, and what Griffith did is he took living S cells. So if you remember the S, they look smooth, right? And so essentially he would inject a mouse, and because they were lethal, the mouse would die. And so this is a control just to make sure that this is how it worked. And you can see here that you can see that capsule. Well, then he took living R cells that, if you notice, don't have the capsule here, injected it in the mouse, and because it doesn't have that capsule, the body can protect itself using the immune system. And so here, this rough strain, right, the little mouse is healthy. And so this essentially is a control too to see that this strain doesn't kill the mouse. The last control we're going to look at that he did was he heat killed the S strain. So basically what he did is he took this guy over here. He heated it up. So think about what happens to us if you bake us. We're going to die. And so essentially he did that. He killed the strain of bacteria. And so essentially what you were doing is you were injecting this mouse with dead bacteria, right? And the mouse didn't die. So he knew that the only way that this bacteria could kill the mouse is if it was alive. If it was dead, it did no harm. So what he did then is a very cool experiment. He took dead S cells, right, plus living R cells. So he took these two guys, right? He mixed these guys together and he injected the mouse. And this is where it gets cool, because you would assume that if you had a bacteria over here that keeps the mouse healthy, and if you had the bacteria here that essentially the mouse was still healthy, that you'd mix these two guys together and that mouse would be healthy. But look what happened to that mouse. The mouse died. And then when they looked at these cells, they noticed that these guys somehow changed into these living S cells were present. So this helped Griffith figure out that the characteristics, right, of the S cell moved into the R cell and turned it into S. This is a process now known as transformation. And basically this proved that characteristics could be moved from one organism to the next. And so if characteristics could be moved, there has to be something there that is responsible for the characteristics. And that thing must be moving from one organism to the next. Very simple, but very cool experiment. So the next question then is, if the ability to secrete a capsule could be moved, what was the biochemical basis of this movement or transformation, right? And so that's when we started trying to figure out what was going on. So you guys tell me, the Griffith experiment proved DNA was replicated in cells by S bacteria. So, doesn't really make much sense, does it? So, this would be false. I don't even know what this really means. The DNA experiment proved that DNA was moved or transformed from basically dead S cells, right, to live R cells. So if you read it and it absolutely doesn't make any sense, chances are I'm just trying to get you. So Avery, MacLeod, and McCarty used a purification method to actually reveal that DNA is a genetic material. This happened in the 1940s. And so they had seen the Griffith experiment. They thought it was really, really interesting. And so what they did is they took only purified DNA from S cells that could essentially transform R cells, right? And so they had to be careful that the purified DNA might also contain traces of other contamination. So they saw this stuff and they're like, okay, we think that this is essentially what's causing this movement. But there could be other things in there that we don't know about. So we're going to add 3 enzymes. So we're going to add what's known as a DNase, an RNase, and a protease. Now remember, we already know that enzymes do what they say. So DNase breaks down DNA. RNase breaks down what? You'd be right if you said RNA. And protease breaks down protein. So we're going to add those in. And when they did, they realized the transformation still happened even when we got rid of RNA and proteins. But when DNA was gotten rid of, nothing happened. So this allowed them to realize that DNA is actually the genetic material. So Avery, MacLeod, and McCarty used a purification method to reveal that DNA is the genetic material. True or false? Hopefully you answered true on that one. So in 1952, Hershey and Chase, after looking at this basically load of research coming out, thought, well, let's see if this is just in this particular case or if this is widespread. Does this go outside of this RNS bacteria? So they studied what are known as T2 viruses, and they infect E. coli. They're also known as bacteriophages or phages. So you can see this picture. They kind of look like little robots to me, right? And this is the electron microscope that shows them. So essentially on the outside here, this blue stuff is basically protein. On the inside here, you get the DNA. And these are kind of scary because they have these little claw things that come down here. They attach to a bacteria, and then they take the DNA here and they inject it into the cells. So what they want to do is they want to look to see if basically DNA is the hereditary information found in not just things like this very specific pneumonia type bacteria, but if you can find it as well in viruses.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
And so essentially all they did was they had two experiments, right? And then in this first one, what they did is they labeled the protein radioactive. So this outside part where it's pink is radioactive. These guys put their DNA inside this bacteria cell, and then essentially more of them get made from inside this bacteria cell. They can basically pull it all apart, right? And then they look to see where the radioactive material is, right? And in this one, they did the exact same thing, and they just used the P32, which is radioactive DNA. And what they found was that in these, right, the radioactivity was up here with the original viruses. It didn't get moved into the new bacteria. So, and that's what would happen. They'd get moved into the new bacteria and make more of these viral phages if it was transported through. So they knew it was not protein because it stayed here. But in this case, the DNA that was radioactive moved in to the actual cell, and that's where they found the radioactivity. So that's how they knew that DNA was the material that was responsible for inheritance in these phage DNA, or these phage organisms. And so that happened in 1952. And that's essentially the Hershey Chase experiment, and that's what established that viral DNA actually enters bacterial cells and is required for the synthesis of new viral particles. So now we have the proof, essentially, that it is across organisms. It's not specific for one, or organisms or entities. So with this in mind, did the Hershey Chase experiment happen in 1951? Nope. Oops, I keep doing that, right? They use radioactive DNA and not radioactive protein to show that proteins are required. You should say nope. They use both, right? Establish that viral DNA enters bacterial cells. Yep. Use bacterial virus known as a bacteriophage. Yep. Bacteria, the experiment used both radioactive DNA and proteins. Yep. So essentially we know that it's C, D, and E. Now let's go quick over structure and what it looks like now that we know DNA is the genetic material. And there are loads of other experiments that show this. I just focused on the ones that are mentioned in your book. Now, nucleic acid essentially fits both DNA and RNA, and that the level of DNA actually starts from very small. So the individual monomer, right, is a nucleotide, and this is a building block, right, monomer of RNA and DNA. That's put together into a strand, so it's a linear polymer strand, which means it has a lot of monomers. Now that goes from just being a long strand to being a curlicue, right? And there's two strands that basically walk around each other like a staircase does, and so that's known as a double helix. Now that helix can condense down and form what are known as chromosomes, and all the chromosomes together form a genome. So that's a complete set of genetic material in an organism. And so this is what it looks like, essentially starting as simple as the nucleotide going into a strand, and then you see it's the double helix here. Now that helix is going to start wrapping around these guys. This is what's going to make a nucleosome, and it's held by proteins. And then that basically condenses down to form what you know as those chromosomes, and then all the chromosomes together make a genome. And so the nucleotides itself, it's divided into three parts. If you remember this, there is a phosphate, there is a sugar, and there's a base. So this right here is a phosphate. This right here is a sugar. And this right here is your base, right? What changes in DNA is your base. And so this is called, it's a ribose sugar. That's what ribose looks like. And it's called deoxy because at this number 2, it actually shows no oxygen. So I said number 2 because on these things we count the carbons. And so each dot here, here, sorry, here, here. Those are all carbons, to let you know. And we always start with the one that's on the base. So this is the 1 carbon, this is a 2, this is a 3, this is the 4, and the one up here is the 5. And so at the 2 carbon here, it's called deoxy because there's not an OH. Now when we go and talk about RNA, it's not a deoxyribose, it's actually a ribose, which means that OH will be there again. So that's where the name comes from. Now, the nitrogenous base, it's called that because there's actually nitrogen in there. And essentially, those have shorthand A, G, C, and T. Right here, A, G, C, and T. And the A stands for adenosine, the G stands for guanine, the C stands for cytosine, and the T stands for thymine. Now, these guys are considered what are known as purines. They're going to be smaller, like these. I lied. I misspoke. They are not going to be, those are going to be double. They're going to have basically 2 rings. And then the pyrimidines, the cytosine and thymine, look like this. I apologize for that. And so, and then we're going to talk about, because certain things base pair together. And so if we look, we can look at one side of a particular piece of DNA, and you can see this is the nucleotide right here, this whole thing. And it's part of a phosphate, a sugar, and a base, right? And so you can see here's guanine, cytosine, adenine, and thymine. And they're called that based on what you see here, right? And so guanine and adenine are purines, and thymine and cytosine are pyrimidines. Okay. If you notice down here that the bottom here is on the 3' and the top is the 5' carbon, you'll always see that. That's how they go. They start 3' and they go up to 5', and it's based essentially on how they're put together, and then when we get to actually replication. We're just going to go by those. So RNA is a form of a nucleotide too. It still has its phosphate, its sugar, and its base here, right? And you notice here, instead of just having a hydrogen there, it actually has the OH. Thus the name ribonucleic acid, right? Because it's actually a ribose sugar. It has A, G, C, and then instead of T, it has uracil. So this guy has not contain thymine at all. Always like to think RNA wants you, right? That's how I remember it. Now, on the bases, when we're looking at them, you can see A, G, C, and U, and essentially on here, if you go back, and if you look at this guy right here, oops. Sorry, I'm going around. If you look at this guy right here, this is thymine, and if you notice, there's a CH3O and then NH here, right? Let's go forward really quick. Here, it looks somewhat the same. Here, though, it's going to have a CH3, and you notice that you have the O here, right? So these guys right here on the side are essentially what changes a little bit. Now, just to let you know, when we have this, that these guys base pair, remember we have 2 strings of DNA that come together, right? And so essentially what happens is the bases base pair with each other. So C and G base pair with each other, and A and U base pair. Now A, if you have DNA, will base pair with T, right? And so, and I like to think, you can think at when you're talking about them working together, and G's and C's kind of look the same. RNA is similar to DNA in that they are both double-stranded, have a ribose sugar, and the same base. True or false? It should be false. First of all, DNA is double-stranded. RNA is single-stranded. DNA has ribose sugar goes with RNA, right? DNA has a deoxy. And the bases aren't the same, right? DNA, you have A, T, C, and G. RNA, you have what? A, U, C, and G, right? So that should help you review that. So when we're talking about base pairing, essentially, we write it, like I said earlier, from this, you go from 5' up here at the top to 3' down here. That's how it's written. And so you can shorthand. So you always write 5' on one side and T, A, C, G, and then 3' on the other side to say the end. This right here forms the backbone. And then these are going to form what's considered the rungs of the ladder. So if you're looking at this, basically, the outside part of a ladder, that's going to be the DNA backbone, which is sugar and phosphate. And on the inside here, the ladders are the bases. And so when we start talking about DNA and what it looks like, we have a lot of people we want to talk about. And what do you think these guys won the Nobel Prize for? So we have Francis Harry Compton Crick, James Watson, and Maurice Wilkins here. What do you think they won for? Not the discovery of DNA, because we already knew DNA was there, right? They won for actually being able to uncover the structure. And you can see them standing for promo photos in front of one of their molecular model kits. Now, we also have Rosalind Franklin. And Rosalind Franklin was very pivotal in the discovery of the structure of DNA. What happened with her? Did she get credit for her discovery? Did you notice that her picture isn't down there? She actually worked in Maurice Wilkins' lab, and she was actually doing research trying to uncover the overall structure of DNA. And this is her X-ray diffraction pattern right there. And this is essentially what happened is Watson and Crick came to her and said, hey, can we look at your data? And she said no. She was working on a bunch of other things at the same time. And so what they did is they went behind her back and asked Maurice Wilkins if they could get it for him or them. And Maurice went and got it, made copies of it, put it back. And then that's what they used behind her back to figure out what the shape of it is. So, yeah, she would have gotten some credit, perhaps, if she wouldn't have died. What did she die of? If you said cancer from the research that she did, you would be correct. So, yes. So, in 1953, James Watson and Francis Crick actually proposed the structured DNA double helix, right? And they had used, because Linus Pauling was working on this too, and you might know the Pauli excursion principle and all of that. He's very big in chemistry. And so, use Pauling's method of this ball and stick modeling. We kind of used it when we were doing the toothpicks and the gumdrops. It was an easy way to do it, but they actually have some that you can buy that give you precise angles for you to put together. And so they were using this, and they used Rosalind Franklin's X-ray diffraction pattern to actually be able to work the two together to come up with a helical structure that had uniform diameter. And you can tell that it has uniform diameter because these guys right here, if you look, these dots are actually showing you the base, and you can see that they go equal distance out. And it looks almost like, you know, you're going through a tunnel there, which is how they knew. And this is also part of it as well up here. And that's essentially how you knew that this was a helical structure. It's coming from the top. And so what they did, or what she did, is she took an X-ray beam and she shot it at this wet DNA sample. To this time, if DNA is not wet, it actually folds a different way. So you have to be careful. And then they basically bounced off, and then they were collected on a photographic plate, and they could basically, the pattern represents the atomic array of these fibers that were in it. So they did the X-ray, her and Maurice Wilkins did the X-ray diffraction analysis on DNA. And then basically Wilkins made that data available to Watson and Crick without her approval. And this is a nice image of what it looks now when you do it, much more detailed than it was back then. And so what they were able to tell, they did know we had these purines and these pyrimidines that I also talked about. And for the longest time, they were putting things in the wrong way. And then for here, they started thinking, well, what if they're in the middle? And the reason why we know, and we're going to talk about Chargaff's rule here in a minute, that he came up and he was able to determine this. How we know that purines and purines don't come together is because when you look at that X-ray diffraction pattern, they're too far away. And pyrimidines, they're too close. So when you have one with the other, that gets us the perfect amount of distance between the two that the X-ray diffraction pattern shows. So let's actually look at what DNA looks like.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
Okay. Let's see. Okay. Sorry about that. So when we look at DNA, we know it's double-stranded. It's anti-parallel, and what that means, do you see how we follow this side here, that you have 5' down here and 3' here? Well, on the opposite, we have 5' up here and 3' down here. So we have 5 to 3 and 3 to 5. That's what it means by anti-parallel. It's right-handed, which means if you take your right hand and make it curl around like you're kind of going in to kind of hug and bring your hand back, that it's going to come up that way, basically. This backbone that's on the outside is made up of sugar and phosphates, and that the bases are here on the inside. What basically stabilizes the 2 strands together here are hydrogen bonds. And the covalent bonds are around here that hold these guys together. Those covalent bonds essentially are called phosphodiester bonds.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the 2 sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
Okay. So who came up with the base pairing? Erwin Chargaff in 1950 analyzed base compositions of DNA from many different species. And then when you looked at the X-ray diffraction pattern, they were actually able to give a more of a definite show. So what he did is he looked at the amount of adenine, thymine, cytosine, and guanine. And when he did the analysis, he realized that there was equal amounts of adenine as there was thymine, and then there was equal amounts of guanine to cytosine. And then when you added this to this, you came up at like 100%. And so you can basically figure out that adenine, since there was equal amounts of adenine as thymine, then they essentially would base pair with each other. And since there was equal amounts of cytosine as there was guanine, and then they would equally pair. For instance, if you looked and said that 20% of the sample was adenine, then you could basically look and then 20% of it would be thymine. And so that equals 40%. So what's left over is 60. And then you could see essentially there was equal amounts of each one of these. So that's how Chargaff was able to basically hypothesize that these two guys were together. And so you can see here that you have these double-stranded coming together, and essentially they base pair really well, especially with them being anti-parallel. They're anti-parallel, so they fit in the same plane. And then you can see that the hydrogen bonds actually hold those two guys together there. So Chargaff's rule basically just states that A base pairs with T and G base pairs with C, and that it keeps it consistent. And when you look at the X-ray diffraction pattern, it shows that the width is consistent with that. And so you can figure out what complementary strand or the second strand you have all the time by using this. And so essentially, if you look at this and you are given this sequence right here, you can figure this one out by using Chargaff's rule. And we're going to do some of that in class. Now, if you notice that if one strand is 5 to 3, then it is an anti-parallel, so the other strand is 3 to 5. And so practice this essentially on your own. Keep it, and then we're going to talk about this when we get and continue the discussion on base pairing. Thank you.